Hello everyone, I'm so glad you were able to come to my talk. So as Daniel mentioned, I was a master's student here and then I defended back in July. So this is going to be a little bit of what my thesis defense was, what my project was, as well as what I've been doing since I graduated. So without further ado, fine scale, terrain sampling, and niche modeling of staghorn coral in the lower Florida Keys. Before we can talk about staghorn coral, let's talk a bit about coral reefs. It's a super diverse and valuable ecosystem, not just for the ocean, but also for us land lovers here. The coral reefs are really important towards buffering shorelines against storm surge and flooding events. It's also very important economically when it comes to our commercial fishing and ecotourism industries. But over the last few years, over a fifth of corals have died, or over the last 30 years. And in the coming decades, it's projected that 70 to 90% of corals will decline as we approach 1.5 degrees Celsius of warming. Here you can see three corals that are especially important towards our southeastern oceans: the staghorn coral, elkhorn coral, and star boulder coral. Focusing in on staghorn coral, they are a stony reef-building coral that is found throughout the Caribbean, Bahamas, and Gulf of Mexico. Here you can see in this image, the pink is their shallow water distribution and the light blue is their deeper distribution. They are fast growing and long lived and are very important towards building the reef. Without them, the reef system doesn't exist. They've been labeled critically endangered, and there's a few different reasons for their decline. First off is high intensity thermal events. I don't know if anyone was paying attention towards the Florida Keys over this past summer, but the ocean temperatures were reaching record highs over 100 degrees Fahrenheit, and these sort of bleaching events, is what they're called, can cause mass mortality of staghorn corals. The bleaching is because these staghorn corals and corals in general have algae that live in the organism. It gives it this bright orange color, and these high temperatures cause the algae to leave, and it turns this bright white color, ergo bleaching. So we have these really devastating intermittent events, and then more continuously we have disease. Here you can see an example of a staghorn coral that's been infected with disease. On the outer side there are these orange tissue, healthy, and then disease is slowly encroaching from the center. There's a few different diseases that affect staghorn coral. Throughout the Florida Keys, the main three are black band disease, white band disease, and Caribbean ciliate infection. There's also been pollution and runoff from our human settlements, and another cause for their decline is corallivores. Here you can see two examples of them. Corallivores are organisms which eat corals, corallivore. We have the damselfish, which is this really aggressive territorial fish. When I'm in the water, the little guy's trying to bite me and get me away from the coral. We also have these beautiful parrotfish that, especially for young corals, can just eat the entire organism, and both of these can eat it, and when they chew on the organism, it can cause these lesions, which disease can thereby enter into the organism. So because of this, there's been a suite of restoration efforts to help restore their populations. A major form of this is coral outplanting. This is where young corals are reared in the lab on land and then are planted out to the environment. Here you can see it's sort of an in-between stage. After they are taken from the lab, they're put on boats, and then they are put on these coral trees. This is to help get them used to the marine environment, the temperature fluctuations, all the stuff in the water. These corals are elkhorn corals, which are a very cousin to staghorn corals. And then afterwards, they are planted onto the reef. Complementing this are genetic improvements. We can genetically select or manipulate these staghorn corals to become more resilient to marine heat waves or disease. We can also use antibiotics to help fight off disease. We have built physical structures to discourage corallivores, and what I find particularly interesting are habitat suitability models. Habitat suitability models use environmental and biological surveys to project where organisms are best suited to survive. This can take multiple forms. We can understand where they're going to occur, where there's going to be higher abundances. We can also better understand range expansion. So maybe not where corals are best suited now, but as ocean temperatures increase, where they might be able to better survive in the future. We can also model where they're best able to grow or higher fecundity, which is them reproducing. So it's a really helpful tool towards understanding where organisms will be, but it's also really helpful towards understanding ecology and the way that an animal interacts with its environment. We can do a habitat suitability model, and if we're seeing that it doesn't really match the reality of what's going on in the environment, we know we're missing something. So it's really good for troubleshooting our understanding of the ecosystem. So through all of this, I became interested in answering the question, can habitat suitability models support outplantation efforts? There's been some habitat suitability models developed for staghorn coral. This one was done by Van Woesik in 2020. They did habitat suitability models for a bunch of different organisms. And from their results, they saw that staghorn corals do better in the upper and lower Florida reef tract, here you can see in the red boxes, on areas of the forereef. They did not so good in the Middle Keys due to the negative effects from Florida Bay. I would like to be away from Miami as well. So we also see that the staghorn coral does better in areas of moderate sea surface temperature range. The coral's very sensitive to high temperatures, but also a lot of fluctuations. So moderate sea surface temperature range, moderate sea surface temperature mean, and we also see that they do better in areas of moderate flow. This flow helps to provide more food, provide more oxygen, and helps keep the water moving. We also saw that the coral did better in turbid waters. The turbidity helps to mitigate temperature fluctuations and UV radiation. There are, however, a few issues when it comes to using these habitat suitability models. When talking with restoration workers, they said that they knew about these models, but due to the coarse resolution of the data used, it results in these recommendation sites that are really large, so not very specific, and also really far away. So it's not economically and logistically feasible to outplant and recurringly monitor. Secondly, what restoration groups are focusing more on is within a given site, growth and survivability can vary really widely. In one area, you can see a staghorn coral that is thriving, growing really well, and a few meters off, you could see one completely decimated by disease. And they're curious in understanding more these fine-scale variances. Another thing that I saw was that these models use wild coral populations. And like I said, they're grown on the lab. They have to get used to the marine environment. We know that they perform differently than wild coral populations. And then lastly, not just with habitat suitability models, but with outplantation studies in general, there's been very few long-term studies. Most of the research is done in the first few months to maybe the first year of outplantation. And this is just not really helpful towards understanding how to build long-sustaining reefs. This has been recognized, and there has been a few long-term studies, and of the long-term studies that have been done, it was shown that growth and survivability decreased after 2 years. So from all of this, I had the three main questions for my thesis. One, what are the coral cover and growth trends in the years following outplantation? What do we see? Do we see those 2-year dramatic decreases to be occurring everywhere, or was it just that study? Next, what are the fine-scale seafloor and terrain characteristics that are attributed with the variation in healthy coral cover and growth? Answering that question, why is this coral doing okay and this one a few meters off not? And then lastly, I wanted to understand how average annual high-intensity events influence these outplanted corals in their growth and survivability. So, looking at data collection, as some of you might know, within the Spatial Ecology Lab and Dr. Dykstra and I, we've been going down to the Florida Keys in the last 2 weeks of June. And so this is going into a bit of what we're doing down there. So we've been working with Mote Marine Laboratory. They've been outplanting staghorn coral for a really long time now, and we surveyed 8 of their outplant sites in the lower Florida Keys, 4 of which are in sanctuary preservation areas, S4 and S6, T3, and T16, down in that southern part, and then the other sites. We surveyed all of these sites in 2022 and additionally surveyed ICC-1 in 2021. All of these sites, except for ADAC-1, are in the forereef, and of course, all of these are in the lower Florida reef tract. So hypothetically, from that Van Woesik suitability model, this is the best place for the staghorn corals to be in. For time outplanted, our youngest outplants were in ADAC-1. They were a few months out before we went out to survey, and our oldest outplant site was in T3, where they had been outplanted in 2017, so 5-year-old outplants. Here you can see an example of our outplant site from an aerial surveillance and then what it looks like in the water. We have these elevated coral reef areas where the corals and the fishes are, and they're cut through by these current-driven channels. When I'm in the water, it kind of reminds me of this scene from Finding Nemo. Beautiful reef in the background and then assured death. Looking at what it's like in the water, here you can see Dr. Dykstra is collecting images. We have this big rig called the Monster. It has 2 Canon DSLR cameras in a waterproof housing, and we collect images in this lateral and horizontal lawnmower pattern. And here I have a video. So this is what Dr. Dykstra is doing. Meanwhile, I'm swimming around collecting a bunch of other data. We collect data at our 10 by 30 or 10 by 60 meter ponds. So that's what we do down in the Florida Keys. After that, we come on back home to UNH. This is where we start doing our image processing. The photos are uploaded into Agisoft Metashape. This tile is 10 centimeters by 10 centimeters, and this site is a 10 by 30 meter. The tiles help us to stitch the photos together within the software. So this is one of our orthomosaics, ortho-rectified photo mosaic. Here's a larger image. We also, because the images are taken at multiple angles, are able to create 3D models. So here we're taking a more lateral view of that Staghorn coral. And then because we're creating 3D models, we can also create digital elevation models. And hopefully you guys will be able to see, the color can be a little off. From this, we can see elevation just from the corals themselves, the rocks, and we can get a really good detailed view of the digital elevation in this site. So that's an Agisoft Metashape, and from there, I was able to collect my additional data from ArcGIS Pro. So breaking down my explanatory variables for my model, they can be grouped into 3 main forms. The first is terrain attributes, which is depth, slope, aspect, curvature, and roughness, and this is all from the DEM. Additionally, I wanted to collect some other environmental characteristics and then high-intensity events. I'll get to those in a minute. Looking at the terrain attributes, our DEMs were in the scale of millimeters. Our coarsest one was 8 millimeters, so I resampled them all to 8 millimeters. And then taking those and our orthomosaics, I imported them into ArcGIS Pro. I quantified coral cover from using polygons. So I made these polygons to overlay the healthy tissue cover and the unhealthy tissue cover, the healthy being this dark pink and the unhealthy being the lighter pink. I then created 10 centimeter buffers around each of the polygons, and from there I extracted depth, we all know what that is, slope being the degree of incline of the benthic environment, aspect being the cardinal direction that slope is facing. Surface relief ratio refers to how much of the surrounding cells are changing. So a low surface relief ratio means there isn't a lot of change, and a high means that the surrounding cells are changing in value a lot, a very complex looking environment. Profile curvature refers to how convex and concave those changes are. Looking at the other environmental characteristics, these were just things that I was interested in or thought could have an influence. There was distance from other staghorn corals. There was also distance from the reef edge, so that drop-off I was talking about, and then also distance from the coast. I wanted to include this because other habitat suitability models for other corals showed it to be significant. And then lastly, collecting high-intensity events. I collected data from NOAA buoys in Sand Key, Sombrero Key, and Key West. I wanted to collect data on marine heat waves, marine cold waves, and high wind events. I used this definition from Hobday et al. For what a marine heat wave is, which is 5 continuous days where the average daily temperature for that day exceeds the 90th percentile based on a 30-year historical baseline. So had to get that 30-year historical baseline. I was able to collect temperature data from these buoys all the way back till 1988. So that was my definition for marine heat waves. I also use the same definition for marine cold waves, where temperatures are going below the 10th percentile from a 30-year historical baseline period. High wind events I defined by days where the average winds exceeded 12.78 knots, which is the average sustained wind speed from a tropical storm. I wanted to avoid collinearity with time, and also I wanted to capture the intensity and duration of these events. So I transformed the data to be the intensity, how much it goes above or below that threshold of mine, divided by the duration, how long the event had occurred, and then divided for the number of years a coral had been outplanted for. So that's my explanatory variables. Looking at the response variables, I had three main ones. First is percent healthy coral cover, and this refers to the percentage of a single coral outplant that has healthy tissue cover. So 100% healthy means that it has no disease, no bleaching, no nothing, it's completely healthy. And 0% means it seems to be completely bleached, completely covered by disease, there is no healthy tissue cover and is dead. I also wanted to include total growth, which is the change in centimeters squared per year of the total coral skeletal size. So this is including bleached and diseased coral cover. And then I also wanted to include healthy growth, the change in healthy coral tissue. I included this because I wanted to see where, despite having been infected by disease, a coral is continuing to grow and produce healthy tissue. After excluding fragments and the wild coral population, I had about 1,168 corals for my model. All right, moving on to data analysis. I'm not going to belabor the statistics. No one wants to hear me talk about that, except for maybe Kim. So we have three main models that I used. One for total growth, one for healthy growth, and one for percent healthy cover. I also decided that I wanted to run sub-models. Sub-models are like regular models, but smaller. They essentially are subsetted by whatever. I decided I wanted to have them subset by age. So if I saw any changes in corals and their behavior over time, I wanted to see how young corals versus older corals responded to these explanatory variables. Model selection took the form of excluding highly correlating variables. This didn't come into play for my overarching models, but it did come into play for my submodels. Moving on to the results, that first question, what are the variations over time? Here you can see that for percent healthy coral cover, in the first couple of years, we're seeing around 100%. Corals are doing well. They're keeping themselves mostly healthy, except for maybe a few outliers. And then after 2 years, we see this dramatic decrease. We start to see corals completely covered by disease or bleached, and we see this median value really dropping down. So a few things. One, this emphasizes the fact that we need to do long-term studies. We can't just focus on the first 2 years, otherwise we'll pat ourselves on the back and say, we've done great, guys, we can just ignore it. We have to focus on looking at long-term outplants. And like I said, I wanted to run submodels. So from this, I ran a submodel for corals less than 2 years old and older than 2 years old. I'll just say right now, when it comes to the submodel for corals less than 2 years old, I found no significant results. Nothing could tell me where corals less than 2 years old did better or did worse. There were results for corals older than 2 years old, and I'll get to those in a minute. But looking at the model results for all of my corals, we saw that corals did better farther away from the coast. Like I said, other corals have shown these results in their models, so this wasn't necessarily surprising. And it has likely been attributed for other corals to be farther away from anthropogenic influence, that runoff and that pollution. Another reason possibly for why I got these model results is that the corals that are farther away from the coast, this S6, S4, T3, T16, and M32, they're also farther away from the middle keys, which we know are pretty bad for staghorn coral, so it could just be a confounding of those two variables interacting. Next, looking at distance from the reef edge, it showed that staghorn corals did better when they were closer to the reef edge. And this is likely due to the fact that the reef edge is where the water is flowing through. There are these current-driven channels, so there is a higher water flow occurring near the reef edge. These results were observed by Mote Marine Laboratory and has caused them to already start preferentially outplanting near the reef edge. Another result that was shown to be significant, not just for all of my corals, but also for the corals older than 2 years old, was depth. When it comes to healthy coral cover, they did better in deeper waters. And this isn't surprising. When you go deeper, you're going to have less UV radiation and temperature fluctuations. Another result that should be significant for older corals and all of the corals in general was roughness. Now, as you can see, it's very variable in the fine scale, so zooming in a little bit, we can see that high roughness, which is the sort of redder colors, is indicative of more soft coral, more rocks, more stuff going on, while less roughness is indicative of having more space, more sandy bits, more flat. For my model results, it showed that staghorn corals did better in less rough terrain. There's a few reasons, possible few reasons for this. First, being near other corals and more stuff going on could be helping to cause this density-driven disease, spread of disease. So as we know from our time in COVID, that being around other people can help infect us. So these results possibly are saying that we need to have our staghorn corals socially distanced from other corals. Another thing could be that when you are surrounded by the areas where there are more corals and more stuff going on, where the damselfish, where the parrotfish, where other corallivores want to be. They want to be where all the other food is. So if they're in more complex terrain, putting corals farther away from complex terrain could create some space from the corallivores that want to chew and introduce disease to the coral. And lastly, this could also just be plain old competition, with staghorn corals being surrounded by other organisms that want to get the food, get the oxygen that the staghorn coral wants. Having them being spatially distant could mean that there's less competition. All right, percent healthy coral cover, done. Moving on to growth, there's a few things to notice. First, we see that when it comes to total and healthy growth, for both this image and also my models, there wasn't much of a difference between the two. So when I start talking about growth, I'm talking about both. Next, we see that unlike percent healthy coral cover, we don't see this dramatic decrease after 2 years. For the most part, growth is staying above 0 centimeters squared per year, so they are continuing to grow. And yeah, there isn't a dramatic decrease after any period of time, so I didn't run sub-models for growth. So we saw corals did better when, in growth when being farther away from the coast and in less rough terrain, which is the same for percent healthy coral cover, likely due to the same reasons. Staghorn corals grew better in areas of a high slope relief, but did not show to grow better near the reef edge, or the models were showing that. So the reason possibly for this is that when it comes to the reef edge, this is a definition I came up with. There is no like hard term for what a reef edge is. It's just what I saw. And so when looking at this image, the reef edge sometimes has this really starkly defined drop off, and in other times can peter out a little bit. We see that when there's a higher slope, there's more constrained water flow. From that current channel, the water is being stuck in that one channel and it's going forward. But when the reef edge kind of becomes less steep and it kind of peters out a bit more, the water is able to disperse more. From this, we might be able to say that when it comes to percent healthy coral cover, the corals want to be where the water flow is, but the degree of growth is dependent upon how much water flow is happening near these more steeper drop-offs. Next, looking at curvature, like roughness, very variable in the fine scale. So zooming in a little bit, convex terrain is referring to similarly roughness where stuff is sticking up, while concave environments are where it's smoother and there's not as much going on. So we see that staghorn corals grew better in more concave environments than instead of convex environments, likely for the same reasons I mentioned for roughness. So second part, done. Answering that third question, how do high intensity events influence staghorn corals? So you might be surprised to see when looking at these results, so positive coefficient mean it's helpful and negative coefficient mean that it's not helpful. High wind events showed to be positive for both total and healthy growth. This is a bit surprising to me. When I first saw this, from what we know about high wind events, they cause pollution, they cause destruction, they go over human settlements and wash stuff out to shore. If they go over the reefs, they're breaking off staghorn coral, can allow disease to therefore enter into the organism. But high wind events are not always bad, especially when it comes to interrupting bleaching events. When a hurricane or tropical storm comes through, it helps cool the ocean temperatures and can stop bleaching from occurring. It also helps with wave action, more mixing, more food, more oxygen. So from this, it's also important to note, my threshold for what a high wind event is a bit lower than what other people might have used. So from these results, we might be able to say that moderately high wind events are not always bad. When there's enough wind that causes cooling and mixing, it's helpful, and then it's bad when it goes directly over human settlement, directly over the corals. Next, my model results showed that marine heat waves are bad. Who knew that? Certainly not I. So there's a long-standing history marine heat waves are bad for stag or corals in general, not just Staghorn corals. But what might be surprising to see is that marine cold waves showed to be more bad, or just as bad as, than marine heat waves. We don't really think about marine cold waves very much. As climate change is continuing, oceans are warming, marine cold waves are not what we're concerned about. But when they do occur, they can be just as devastating as marine heat waves. Back in 2010, a cold water event and caused the same amount of bleaching and death as marine heat waves. This is important to consider that as we, as I mentioned, are genetically influencing corals to become more resilient to marine heat waves, that we don't accidentally make them more vulnerable to marine cold waves. We need to maintain or possibly even increase their resilience towards marine cold waves that, while rare, will still occur in the future. So looking forward, I hope to use these results with Mote Marine Laboratory to understand where within their current restoration sites are the best places for them to outplant so we can build long-standing healthy reefs for the future. That was my thesis defense, yay! I had a really good experience during my master's, got to do a lot of fun scuba diving, swimming stuff down in the Florida Keys. And thankfully, I was lucky enough to get hired on and continue on with this work here at CCOM. And it allowed me the opportunity to sort of follow up on some of the stuff that I was like, I kind of am curious about that, but I'm doing a master's and not a PhD, so we'll just ignore it for now. Looking forward, I became interested in terrain attributes. Now, like I said, we were working at the millimeter scale. Most habitat suitability modeling is at the meter to kilometer scale. So when it came to sampling methods and the ways I was going about my models, I was using methodologies that other people were using, and I found it doesn't really translate when you start looking so fine scale. Terrain attributes are computationally dependent. It depends on how you compute them. So you can, within depth, slope, and aspect, you have a few different forms of calculating it. And then curvature and rugosity and roughness, this is just within ArcGIS Pro. There's other versions elsewhere. So I was using what I saw in the literature, but I'm kind of curious, what do these other terrain attributes do? What do they look like? And is it maybe more helpful? Terrain attributes are also resolution dependent. The values we collect at 1 millimeter are going to be different than 1 kilometer. So is 1 millimeter the best? General rule of thumb is finer resolution is better, but when you get into finer resolution, you can start having noise and artifacts influence your results. Next, cell calculation windows. So this refers to the number of surrounding cells that are used to calculate the center cell. So a 3x3 cell window is just going to use the immediate surrounding cells, and then you can go farther out. When you're working at a kilometer scale, you don't really need to go super far out. You can use the immediate surrounding cells, but when you're working at a millimeter scale, if you use the immediate surrounding cells, you're going to amplify the very small variances. A slope of this, but at a millimeter scale. So is that helpful when looking at fine scale, or are we introducing too much noise, too much extra stuff? Terrain attributes are also platform dependent. So all of this was based off of stereo imagery from the ground tree. But in the Florida Keys, we've been also collecting unmanned aerial surveillance and multibeam. If I use this instead, would the model results be different? So terrain attributes are platform dependent. This all begs the question, do these different terrain attribute sampling methods influence habitat suitability model results? If so, how much? And what's the best one? I kind of answered a little bit of this question during my thesis. When I started off, I was collecting slope, curvature, and aspect from the surface information tool, curvature tool, and aspect tool. And I started getting really extreme values that I knew didn't make sense. A few months before I had even started my master's thesis, a new surface parameter tool had been released, and this was made specifically for fine-scale terrain attribute sampling. First, it fits a surface instead of a plane to these cells, and it also uses larger cell window calculation sizes. This causes less extreme values, especially at the finer scale. So taking a look at the differences between these two, we can see that when fitting a surface to cells, that's the new tool, we get slope values that are a lot more reasonable. 0 slope to 60, 70, 80 degrees at the highest. When fitting a plane to a cell, it's mostly 90, 80 degrees incline, which I know that's not true, and no values below 20 degrees incline. So I knew that was wrong. With curvature, the median values are the same for both tools, but when fitting a plane to cells, we get these really extreme outliers, which again, I know don't make sense. When it comes to model results, we see that when using the old tool that fits a plane to the cells, curvature was shown to be more significant, or was shown to be significant for corals older than 2 years old for healthy cover. And when it comes to growth, distance from coast, roughness, and high wind events stopped showing to be significant. So obviously our sampling methods matter when it comes to habitat suitability models. All of this led me to work on this new project, Optimal Terrain Attribute Sampling Methods for Habitat Suitability Modeling in the Fine Scale, which I can talk about at another date. I will be presenting this work at the GeoHab conference in a couple of months. I do have some preliminary results that I won't go into now, that if you want to talk about more, I would love to do so. But for now, that's been what I've been working on, and I would love to take any questions. Yeah, Haley?
Thank you so much. So I'm just kind of curious, like when I'm not as familiar, but when you're looking at lab-grown corals, you have to choose a substrate, I'm assuming, to anchor them and then put them in the wild. I guess I just don't know. What do corals take to in the wild? What do you use when you're doing a lab-based coral? And does that change at all if like sediment types change in that region that you're looking at? Maybe the sediment types are kind of explained by roughness, but
yeah, so
substrate, yeah.
So in the first part of your question about like what we're using, part of it depends on the corals. So Staghorn and Elkhorn coral are these branching corals, and then the other coral that's been outplanted by Mote Marine Laboratory is star boulder coral, which is a mounding coral, and those use these sort of plugs that they have like a little base that they stick them on with some glue, and then they just are able to plaster of Paris it to the environment. When it comes to Staghorn coral, you've got to kind of take in a little bit of a branching material and you got to get it to stick correctly upwards. So there's some cable ties, some nails, and plaster of Paris used to stick them to the marine environment. We also want to make sure that we don't have anything come through and sweep them away, although that does happen. So for that, that's what they've been doing. When it comes to sediment type and what they're putting it on, me and my advice supervisor were talking this a little bit about, yesterday. So that's going to additionally be something different than wild coral populations. Larvae are going to stick more to harder stuff than possibly sand, where there's more opportunities for them to get buried and die. And that's not as much of a concern when it comes to outplanting. I'm not entirely sure what they've been preferring to outplant to. I know that when I'm in the water, I especially see them on more harder surfaces. Yes, Ken?
Hi, I have a math question. I apologize in advance. Any of you can answer it. It's more like a clarification question for myself. When you're talking about fitting a surface to a cell,
Yes.
and you use the distinction between like a plane and a surface, is that like a linear function versus a quadratic function that you're fitting? And what exactly are you fitting it to? Like the slope, the curve?
I actually would love to use you to help answer that question, because fitting a plane versus fitting a surface is what the website told me. And I saw that and I was like, what does that mean? I don't know. I think from my understanding, it might just be fitting a plane as being more linear. while fitting a surface is not. So I honestly don't know, and to be honest, going through the literature kind of went beyond my understanding of what they were talking about. But yeah, I just knew that fitting a surface allows for things to be more dynamic. How? I don't know. Cool. This is why I shouldn't be asking math questions.
Sorry, sorry.
Yes, Harley?
So, you showed that marine cold events actually seem to have more of a negative effect on the corals than marine heat waves.
Yeah.
Is that because of the swing in temperatures? Because marine cold waves are a more dramatic swing in temperatures. If the general cooling trend happened over a longer period of time, do you think there would be more resilience in the corals that you would see?
I'm not sure. I'm trying to go back a few years to see what those marine cold waves look like on the graph. I don't remember, like I said, it's based off of a 30-year historical baseline, which by the way, that means it's going to shift upwards as time goes forward. So the threshold is based off of a historical period. I don't think there's as much of a swing when it becomes cold. I think the swing goes up or down. So marine heat waves happen during the summer, marine cold waves can happen during the winter. So I don't think there's as much of a swing. I do know that Staghorn corals are like one of the most sensitive corals towards temperature fluctuations. So yeah, I don't, and actually I think that was one of some of the questions at my thesis defense, being like, why do we see them to be more sensitive towards cold waves? And I think the answer is we don't entirely know. And also people don't really care. Like I said, we're focusing on heat waves. There's not a lot of studies on cold waves.
Follow-on question. Your definition of a marine, you said your definition of a marine marine cold wave was somewhat higher or lower than, or was that that was the wind events? That was the wind events.
Yes, yes. Marine heat waves, the definition I took from a paper that was the 90th percentile. I didn't see a definition for marine cold waves, so I was like, let's just flip it. So it's anything below the 10th percentile.
Is there a time frame associated with that, or
30-year.
okay?
So yeah, from a 30-year historical baseline, and each day, March 9th has a 90th percentile, March 2nd has a 90th percentile, and March 3rd has a 90th percentile. So each day has a specific degree threshold.
Got it, thank you.
Natalie first, and then you.
I was wondering what, I guess, the ideal temperature range is for the Staghorn corals, as well as how much that overlaps with the coral wars that you were talking about.
Yeah, so first off, cooler than now, but there isn't like one set temperature. A lot of organisms depend on temperature fluctuations inform when certain things are gonna happen. Like reducing larvae, that happens at a certain temperature, it happens seasonally, so those temperature fluctuations aren't necessary. So they like the seasons. In terms of like one set one, I would have to double check, but essentially what the temperatures were before 1970s would be a good baseline.
Thanks for a nice talk. Seems like this habitat suitability modeling can be used to help us focus our efforts to be more effective. I guess that's the idea, but I'm wondering, sort of stepping back, is this kind of thing tractable, or not saying we shouldn't be doing it, but like, can we do this on a scale that makes a difference? Is anybody looking at that? Do you have any thoughts about that?
Making a difference like with outplantation?
Yeah, to do this restoration.
Yeah, so we have these broader scale habitat suitability models that, I mean, I could be wrong, I didn't talk to Van Moosick, but it doesn't seem like they might have been talking with restoration workers when it comes to what was helpful. And then I was talking with restoration work and I went real fine scale. I swung the other way towards really focusing in. Trying to find a nice intermediate. The Moat Marine Laboratory has been focusing on using drone unmanned surveillance imagery. So kind of taking a bit more of a broader image, and they're hoping to use that to inform where outplantation should occur. So that's sort of like the right now. When it comes to the future, it seems like the future is going deeper and farther away from the coast. So that's a conflict of abilities. We are not able to go out physically in order to do surveillances when it's super far out from the coast and deeper. So there needs to be more work, technologically speaking, towards surveillance and modeling. And something that I've been talking with Gabby Trudeau on, her PhD, part of it is using neural networks for finding corals from satellite imagery. So that way we don't have to go out physically to do monitoring, and we can also do monitoring through satellite imagery. So when it comes to like what's fixing things, like outplanting's been done. The marine heat waves this past summer killed so many of our outplants. So we can continue working, but outplanting and habitat suitability modeling only works so well within what we're physically able to do. We kind of need to start doing some technological developments towards going farther away from coast, and habitat suitability modeling can help that out. If that makes sense or answers your question.
So the broad idea is, can we do enough of it fast enough to keep up?
Oh, that's always the question, and hopefully human innovation will always step up to the challenge. But yeah, we definitely need to start being a bit more innovative, because currently what we're working on, ocean temperatures are just increasing too much, that where we're currently restoring stuff, it's not tracking at the same rate as heat waves. So yeah, not to be discouraging. Yeah, Mike?
Adding on to that, in areas where you've seen the populations decline or be displaced, I guess, is there anything filling the ecological niche that it occupies, or is it just kind of a
So like,
barren?
so the corals are known as a keystone species. That means that without them, they define the reef system, and they define it because their body is hard and provides structure. Soft corals have shown to not be as vulnerable to marine heat waves, but they are not keystone species. They're not able to define the environment. So I do know that we've tried to manufacture reefs. Nature does what it does best. We have not really been able to do the same thing as corals themselves are. So we're trying, but answer no.
Yeah, so I guess I was curious about when you're determining roughness of your topography, I guess, or your photographs, are you able to filter out the existing coral, or is that considered part of the environment? You just mentioned it being a keystone, so is that?
So I did include the photogrammetry did include the organisms, so it did include like the roughness and convexity was coming from soft corals, which again, why it wouldn't be necessarily translatable to other platforms like multi-beam and other stuff. So yes, we did include it. There are options to not include it, and that is another thing that I sort of was wanting to follow up on, but haven't done that yet. Yeah.
Since nobody has asked a fluids question yet, I have to ask one. So it's probably ultimately a question about nutrient transport, but how similar are the flow conditions at your different sites you're tracking from a wave resource and current perspective? This maybe relates to flow scales and your metrics of curvature, roughness, size, and all that. Or are these nutrient uptake processes just on a very different time scale, so it doesn't really matter what you're, sort of two questions in one, I guess.
Yeah, so wanted to include that when it comes to food and like nutrients and stuff like that, wanted to include that required certain materials of measurement that we didn't do. When it comes to looking at water flow, that's actually what we're working on now. We have tilt meters to try and collect water flow, and we're trying to get some more finer scale details when it comes to that. We've been deploying this last summer tilt meters by the reef edge and farther away from the reef edge, so we can get a better view of what water flow looks like. When it comes to stuff like nutrients and food, didn't have the tools to measure that. Really wish I could have.
Tilt meters are a nice idea because you can measure both waves and currents, right? So it doesn't.
Yeah, I mean, I am not a physical oceanographer, so looking at that sort of stuff is not my forte. It would be nice to bring some more expertise into the project. Jen's brain, the cogs are turning, she's thinking about stuff.
Thank you so much.